Right, good afternoon, everyone. Can you all hear me well? So I hope you still have some energy left in you to embrace this final presentation for our Science in the News day-long conference. So my name is Saida Tena, and I am a third-year PhD student here at Harvard University in the Division of Medical Sciences. And I have chosen to talk to you about genetic engineering and how that is shaping the future of medicine. Now, when I was looking to put this talk together, genetic engineering is so broad, so I didn't know how much to cover because I could spend all day talking about genetic engineering and its applications. So I decided to focus on two subjects that are really creating a lot of news buzz in the internet and journals, and not only because they carry the implications for really changing the future of medicine, but there's a lot of ethical concerns about these two subjects. So I have decided to talk to you today, so for the next 45 minutes or so, we will cover xenotransplantation. I'm sure this name is pretty foreign to most of you because what is xenotransplantation? And why do we need it? And what are the obstacles for xenotransplantation? And how is the genetic engineering of the technology improving the field? And most importantly, what are the ethical concerns about xenotransplantation? Secondly, genetic modification of the human embryos. Now, I understand that this subject is also very delicate, but also very important to discuss because transplantation, xenotransplantation, and genetic modification of embryos, both of them, they are tied together. So you will see that through my presentation why both of them are important to discuss. So in genetic modification of human embryos, we'll also talk about why do we do it, the obstacles, and most importantly, the ethical issues surrounding this subject as well. So I always feel that as a scientist, our job is not only to contribute to new findings for the future of medicine, but also to present it to the public so that you can also understand it, embrace it. So I'm hoping that by the end of this presentation, we will engage in a discussion because I really want your feedback. You know, how did you perceive this presentation? Is it really important? And most importantly, the ethical issues. You know, what do you think about this? So what your thoughts are? So let's get started. I would like to draw your attention now to the human body. Now, it's not that every day we think about our human bodies and how important it is for us, for all the organs in the human body to work together coherently so that we can carry on our daily lives. So we're all very busy and we don't think about that. But inside of us, there is this big world of cells and organs that work together so they can help us to be alive and also carry everything that we do in our daily lives. But every organ in the body is composed of its own cells. So every cell is important for the function of the liver, for example, is important for the function of the lung, the heart, kidney, and etc. So it is estimated that the human body has approximately 10 trillion cells. Now that's a lot of cells. And all of those cells, they work day and night together so that everything goes smoothly in the organ function. Now within those cells, we have 25,000 genes. And if we were to zoom in into the cell, these genes are within a structure in the cell known as the nucleus. And then in the nucleus, we have these X-like structures, which are called the chromosomes. And that's where the DNA resides. So that's the blueprint of life, DNA. So each chromosome, if you can unwind it, you will find it that is composed of genes. So these genes are the ones that carry the important functions of the cell. They make proteins or molecules that makes us who we are, and then make sure that all the proteins that are expressed, they contribute to a functional organ. But as all these cells are copying the DNA so they can make the proteins that are necessary for each of the organs to function properly, mistakes can happen. And how does the mistake happen? So as we are copying the DNA all the time, if you can see here, this is the double helix structure of the DNA. And within the double helix, the DNA has 4 base pairs. It's A, T, C, and G. Now, they have been paired to work together, such as A and T goes together, and the C and G goes together. But during the copying of the DNA, if the cell makes a mistake, then you can have a mispair, which is, for example, the T that was supposed to have the A is now paired with a C. Now, sometimes this might not be important, and this is going to be fine. But what happens if this mutation now occurs within a gene that is making a protein that's important for the organ to function? Then a mistake is going to happen, leading to organ failure. So organ failure doesn't happen, immediately, but it's an accumulation of time. So for genetically inherited mutations, so when you have mutations that you carry either from your father or from your mother, then they're going to manifest early on within your 20s or maybe later as an organ failure. And these organ failures can be seen in lungs, in the heart, liver, and the kidneys. So these are the genetic inherited mutations that lead to organ failures. But there are also causes that lead to organ failure. So these are diabetes. Diabetes also can lead, if it's not properly controlled, can lead to organ failure, primarily affecting the kidney. Hypertension, the heart and the kidney. Chemotherapy, so a lot of cancers now, they are being treated by very toxic chemicals that also have a lot of side effects affecting our organs that ultimately can also cause organ failure. And also other medications that people take for other diseases. So all of these together lead to organ failure, but the only way that someone can survive when they have an organ failure is through organ transplantation. However, organ transplantation is very much regulated in the United States and also elsewhere in the world. So the only way that a sick person who is listed for organ transplantation is to carry this through this special organization called the United Nations of Organ Sharing that can allocate now the organs that are available for transplantation depending on what the patient needs. But what happens, that this is one of the charts that is taken from the UNOS website, is that as the years go by, the waiting list for an organ transplantation continues to increase, while the numbers of transplant and the available organs for transplantation has remained the same. So there is a great demand for organ transplantation, but there's only so much that we can transplant because of the regulations. So if you go to the UNOS website, you will see that every 10 minutes someone is added on the waiting list. So a lot of people now are waiting for an organ transplantation. However, on average, 20 people die each day in the US waiting for an organ transplant. And currently, as of last night, 125,645 people are listed waiting for an organ transplant. Now, to put this into perspective, worldwide, this number is so much higher. So we have a lot of sick people. They need an organ transplant so they can survive, but the availability for the organs is very short. So what can we do? The answer for what most of the scientists have worked on is xenotransplantation. Now, what is xenotransplantation? Xenotransplantation is referred to transplanting an organ from another species into the human. When we do human-to-human transplantation, that is called allotransplantation. But when we do a different species, it's called xenotransplantation. So that's where the name comes from. And for a long time, the pig has been chosen as the perfect donor for saving most of the people that are on the waiting list. And that is because we have a similar organ size and function. So that makes it perfect because we want organs that are physiologically the same as humans, right? And the size is also important. So the miniature swine, which is called the MGH for Massachusetts General Hospital, because most of the studies have been developed at MGH. As a young adult, the miniature swine, it reaches a certain size where all the organs from the pig match perfectly to the humans, physiologically and also the size. And if we can make xenotransplantation work, we will have unlimited supply of organs, and these particular pigs, they can be bred and kept in clean facilities, which can minimize the infections. Now, when we do allotransplantation, so human-to-human transplants, some of the organs cannot be used because, people can be infected from HIV, for example. So even though they can donate their organs, we cannot use them because you're going to transmit that virus to a healthy person and create more problems. So you're going to save his life, but he's also going to be suffering from other complications related to HIV. So with a pig, we will not have that problem because they don't get HIV. So benefits of xenotransplantation, as I mentioned, some of you already, is that xenotransplantation in itself has been studied now for more than 2 decades. So we know already a lot from what the pig genome is and the size and then the organ function, and also about the genes and proteins that are compatible with humans or the ones that are not. So the successful application to human clinical trials has the potential to end the current long waiting list of thousands of lives and save thousands of lives worldwide. And it's also, xenotransplantation, when it goes through human clinical trials, has the potential to end this black example. But when the porcine organs were transplanted into a baboon, so this is the preclinical studies that are carried out in the US and other centers worldwide, it was observed that the GAL antibodies that we have, they bind to this sugar protein on the pig cells, and then they reject the organ because they recognize it as foreign. In a similar way that we recognize bacteria that invade us as foreign, so we try to attack it to eliminate it so it doesn't get us sick. But that's not what we need because we want the organ to survive. So these antibodies are the same, have the same mechanism similar to the blood transfusion blood groups that require matching. So we all have different blood groups. And if someone requires a blood transfusion, then you have heard that they have to be matched. So if an A has to be matched with an A, otherwise if you match with a B, then the antibodies or the proteins circulating are going to attack those cells. So they're going to destroy your red blood cells. So this is a similar mechanism that is happening here with the presence of this sugar. So most of the transplants that was done, rejections or acute rejections of organ failure was seen immediately for both kidney and the hearts when transplanted, when these pigs had the presence of this sugar in their cells. So in order to move xenotransplantation forward, we need to actually delete this gene because otherwise we cannot move into the clinical trials. We cannot carry any more studies in xenotransplantation because the organ is completely rejected within hours. So how do we actually knock out a gene? So we have the pig genome now, and we need to eliminate this sugar that poses a hurdle for xenotransplantation. Well, that was done based on a technology that led to the creation of Dolly the sheep. Now, some of you have already heard it. This was in 1997. It was a revolutionary technology because for the first time, scientists were able to take a cell from another sheep and take an egg from another sheep, remove the DNA from the egg, and injected the DNA from this other sheep cell into the egg, fertilize the egg, and that let those little embryos grow in culture, and then transfer them into a surrogate mother, and that led to the birth of another sheep. So now this is the first time the technology has shown that by using this, which is called now cloning technology, we can create an animal with the genetic modification that we want. So for example, in this case, we took another sheep DNA, put it to a cell from another sheep, and then created the same sheep from the DNA that we wanted. Now, in the pig, for xenotransplantation, this has the major application of deleting the gene that we do not want in the cell and then creating an animal that misses that gene, or we can insert human genes that are not present in the pig genome, so we can create that transplantation-friendly pig organ. So that's exactly what was done. So the pig cell was modified in the laboratory. The gene that makes the protein was deleted, and then through this technology that led to the dolly, the sheep, we now created animals, so pig animals that were born without that sugar. So these are the two papers that really revolutionized the field of xenotransplantation in 2002. So this is the pig that was born from Revivacor, a company in Virginia, missing that sugar. And this is the one from my own laboratory at MGH named Goldie. For the first time, it was shown that we can genetically modify the pig organ, pig tissues, so that they can have the required modification that we want. However, now that we knocked out this gene and we will have organs surviving into non-human primates longer, more complications arise. So before we couldn't know this because the presence of the gall sugar was the major hurdle. But now we found that there are other unknown pig genes that are not compatible with the human. Now in this case, we cannot delete them because they're important for the pig function. So what do? We have to insert the human gene into the pig genome so that it now can express this human protein so that when the organs can transplant it, we don't have that incompatibility anymore. So you're trying to make the function more human. And these genes were found to be very important for bone marrow transplantation, pancreatic islet transplantation, and liver transplantation. Now these two are cellular transplantation, so we're just getting cells transplanted. But pancreatic islet transplantation is now a venue that has been sought after from so many surgeons to cure, especially type 1 diabetes. So this has the potential also for being applied because there's not a lot of islets that you can transplant in kids with type 1 diabetes. So now the same technology was utilized as the one that we just talked about when we deleted the gene. So in this case now, the porcine cells in the laboratory were modified to insert the human gene that we wanted that is not compatible. And through this technology, now we have animals that are born with human gene, so we call these transgenic animals because now they're expressing that, so they're still missing that sugar gene because there's still a problem for us. But now they're expressing, on top of the missing gene, now they're expressing this human gene that are important for xenotransplantation. So this made a very big difference, at least creating these pigs that express the human genes made a really big difference in the xeno bone marrow transplantation, which is a study that I was heavily involved in. So the problem is that the pig cells here that have the protein that's not compatible with the human or the non-human primate, when they're transplanted, they are being eaten up by the porcines, by the baboon cells, because they do not have that genetic correct compatibility, so they recognize them as foreign and they clear them out from the system. But we do not need that. So if we want to do a bone marrow transplant or an islet transplant, in order for that transplantation to be effective, you need these porcine cells to survive long term. So that's indeed, so we identified what the protein that was incompatible here and genetically modified the porcine cells, and we created for the first time a pig now that expresses this particular gene that is important. So this is me now 6 years ago when I first involved creating this special animal, because through many bone marrow transplantation series, we were able to extend now the survival of pig bone marrow cells into non-human primates for a much longer time. So you can see here that before, when the cells don't have this modification, they're very quickly cleared out from the circulation. But when they have the presence of this human gene, you can see here that the percentage of them that survive is so much higher, and then it goes up to 8 days. So this has already made a significant contribution. And many other genetic modifications that have followed that has allowed for most of the organs to survive now from 2 hours into 5 months, 2 years, and 35 days. So this is already a very huge, big step towards success for xenotransplantation. And in order for FDA to approve a clinical trial, so we can do, for example, kidney transplantation, it requires about 6 months of a life-supporting porcine organ into non-human primates without any side effects. So already 2 groups in the US have already exceeded this expectation. They're about now at 8 months with non-human primates surviving successfully with a genetically modified porcine kidney. However, there are some concerns that the FDA keeps raising, and that's very reasonable because even as a scientist, I accept these concerns as well. So even though we have improved the survival, we haven't done anything about the viruses that reside into the pig genome, that potentially when you transplant these organs, they can create a problem for the human recipients. And in order to create animals that are, again, safer now for transplantation, we need to eradicate those viruses from the pig genome. But in the same time, we can also create animals that have multiple transgenes now. So instead of expressing just human gene, they can express 2 or 3, so they can even improve the functionality and survival of the organ. But with the current technology, we can only do 1 modification. And if we wanted to do multiple modification, it will take years, so we'll never get to the clinical trials. However, the CRISPR-Cas9 technology, which is a very recent technology, has allowed us to do very precise modification at a much rapid pace. So you already had heard before this morning about the CRISPR-Cas9 technology. And the way it was discovered was from bacteria. So bacteria has an immune system against evading viruses, just like we do, but their immune system is much more simpler than ours. So they use these 2 proteins called CRISPR-Cas9, which together work to cut out pieces of viruses that invade the bacteria. So the way it works, in a more simplified version, is that there is a genetic piece within the bacteria that encodes this protein, so this RNA protein, this RNA piece that guides this Cas9 to go where the RNA goes into the genome and then cut it out. Now, the Cas9 itself is referred to as the Caesar of the system. So the CRISPR here allows you to go and find the place in the genome that you want to go to, and the Cas9 follows that and cuts it where you want to cut it. So this is really remarkable technology because we never had such precision before. Now with this technology, we can go and cut a gene that we do not want, but when we're cutting at places, we're also making room for inserting a gene that we also want. So we can do all of this modification in the same cell with a very short of time and much more success rates than with the technology that we had before. So I want to highlight here that the pioneers of the CRISPR-Cas9 technology, which is really revolutionized how genetic engineering approach is done today, are Dr. George Church from Harvard, Jennifer Doudna from the UC Berkeley, Dr. Feng Zhang from MIT, and then Emmanuel Carpentier from the Max Planck Institute. So when this technology was published, it created so much buzz because the technology itself is so powerful. We can do so much with it in ways that we never thought we could before. So CRISPR can be used to treat blindness. It can fix muscular dystrophy, cystic fibrosis, so all of these problems that are actually affecting a lot of people worldwide. So the CRISPR itself has been marked as a revolutionary finding to be the genetic game changer. And more recently, Nature has shown this picture here that CRISPR is everywhere. And we can actually apply CRISPR everywhere, from genetically modifying crops to genetically modifying diseases to genetically modifying the human pig genome to create these transplantation-friendly pigs. So the power of this technology is really big. And indeed, that's what happened for xenotransplantation. So where I mentioned that the problems with the viruses that are still present in the pig genome and can pose a problem for us, were eradicated by using this technology by scientists at Harvard Medical School in 62 sites. So that's really, really precise. To find the 62 sites within the pig genome that have these retroviruses, you go there with a CRISPR-Cas9 and you cut them out. And then you use those cells that were modified with the same technology that I showed. And in 2017, they had pigs that were born to be virus-free. Now, this was really another step towards xenotransplantation that really stirred a lot of news, because a lot of people for the first time worldwide that have not heard of xenotransplantation, now they're realizing the importance of it. And most of it is because the CRISPR technology has really made a huge difference for creating these transplantation-friendly pigs that we have sought for so long. And we could not do it before because we had limitations by the technology. But the CRISPR-Cas9 technology has really given us the tools to move forward and also perhaps go quicker into the human clinical trials. So this is the cover of the front page of the Science magazine, which is one of the most prestigious journals that scientists can publish, really underline how powerful this technology is and how it has really helped xenotransplantation. But some of the, again, coming back to the ethical questions about xenotransplantation. So even though now we have these transplantation-friendly pigs, then we still might have concern about some other microorganisms. So another virus that I mentioned was the CMV virus. So now we also, if that does pose a problem, then we also have to genetically modify the pig again so we can eradicate that. The long term, so the FDA will still be hesitant in sometimes, or even people in general will be hesitant, because this is a very new technology, a new application. While the short term we might save someone's life, we don't know how that person is going, whether his life or her life is going to be affected long term. So are there going to be any other complications related to xenotransplantation that we don't know yet, because we don't have that long-term survival yet? Now, certain religions, they may turn down this because of their religious issues, so they might not accept the technology. And animal activists have always been opposing this technology because of why do we really have the right to use the animals for saving human lives? Organ allocation is an issue, so if this technology moves forward, then who has the right, and where would these animals, for example, kept, and who has the right to distribute the organs for the people who will need them? And then public consent. So this is the most important part, because public consent is you guys. If you accept the technology, if you embrace the technology because it will save life, then that's very important. Like, we as a scientist will fail if we don't have the public consent. So even though we're trying to revolutionize and make a huge impact in someone's life, if we don't have the public consent, then we can all move forward, because the people will not accept it, and then we can all move forward to it. So that's why it's very important, and a lot of centers worldwide, they have made this big effort into talking about xenotransplantation. Why is it so important, and how has the technology changed the game of xenotransplantation into creating these friendly animals, so friendly transplantation pigs that can be totally accepted? Because like I mentioned before, this is not completely foreign to us. We've been using pig tissues before for aortic valve replacement and also the insulin. So accepting an organ transplant would not be foreign again. So that was the summary for xenotransplantation. So we talked about what xenotransplantation is, why it was important, what the obstacles initially were, how did we overcome them, what current obstacles lie, and how the CRISPR-Cas9 technology has really helped us into revolutionizing the xenotransplantation itself. And we also touched upon ethical concerns. So in the end, I would also like to know your feedback about xenotransplantation. But now we're going to talk about the genetic modification of human embryos. So we're going to switch gears a bit, but these two subjects are very much related for reasons that I will talk about. So why do it? What the obstacles are, the powerful CRISPR technology, and most importantly, the ethical concerns about this new modification. So the correct application of genetically modifying the human embryos is to correct a known defective gene that leads to an organ failure. So as I mentioned before, when you inherit a mutation from your parents that is affecting the function of the organ, you're going to get organ failure. But most of these carrier families, or families that for a long time, for many generations, know that they carry this genetic defect, we already know what the gene is and what the problem that is causing. So by using genetic modification early on in the human embryos, then we can correct that defect very early on so that you can have healthy babies being born. Now, if this modification is being done in the germ line, so germ lines are the cells that create us, so the sperm and the egg. Upon fertilization, you have an embryo, and then that leads to formation of the fetus, and then healthy babies. But going into places that we have never thought to go before, such as editing the human embryos, is a very intricate, and also it's very difficult, I would say, because you don't know exactly how precise the technology will be. So although you might modify the gene that you want, you might also create another problem. So that's not feasible for going further and creating the fetuses into full term. So we have to regulate that. We have to regulate that. We also have to fully understand how the technology works before we are 100% sure to apply this technology. But most of the edits that have been reported lately in the news have also been for the reason to try and understand the biological processes, or all the biological steps that are involved into creating a fetus after the egg is fertilized, because this has the potential to help many people who actually suffer from some form of infertility. And the way that all of these modifications are done are through the steps of the in vitro fertilization. Now, I'm sure that all of you have heard that in vitro fertilization is one way of helping couples have babies that will normally have infertility issues. So all of these genetic modifications, again, are done before the IVF. So in the steps that already have been studied and already have been accepted in the laboratories that utilize the in vitro fertilization approach. So here I'm just going to show how the modifications are done in the embryos. So we start by having the egg. So here's the egg, and it has its DNA. Then the egg is fertilized by utilizing the donor sperm, and also now we're introducing the genetic components, so the CRISPR components, together with the sperm. So the only step that's different here from what IVF already is using is the introduction of these genetic components right here. And that is important because when the sperm fertilizes the egg, then you will have a lot of divisions that go on. So this CRISPR component will either delete a gene that is not wanted, for example, or is going to replace the gene that was defective with a corrected gene. So here, when you have fertilization now, because the cell has only the nucleic or the DNA from the mother, when it's fertilized, now you're going to have the DNA also from the father, which comes from the sperm. So that's where the correction happens upon fertilization. Then you're going to end up with these 7-cell stage embryo. So this is very, very early on. So again, as I mentioned, this is during the steps that are carried out through the in vivo with the in vitro fertilization. So this is the step when the scientist now can actually identify whether the genetic mutation has been corrected or not. So when they have this powerful sequencing technology that can correctly identify the embryos that have been genetically corrected now, only then can you safely say that now we can move on into the implantation period so we can create healthy babies. But so if this is this technology has improved and we have studied a lot, only then can we say that we are safe to move this into clinical applications. So again, I want to emphasize that all of these are done in compliance with the steps that already have been accepted for in vitro fertilization. So what has been achieved so far? So what have the scientists created? What genetic modification have they created that has really created so much news worldwide and also feared some people from having this technology applied? So in 2015, scientists in China that genetically modified human embryos to cure a disease called beta thalassemia. I will go in more details why beta thalassemia. They reported that 50% of total embryos showed the correction of the defective gene. Now this is pretty powerful because we never thought we could actually go into genetically modifying anything within the sperm and the egg. But having a 50% chance of success, that's pretty powerful. And in 2017, scientists from here in Oregon Health Sciences University reported embryo modifications in the gene that causes hypertrophic cardiomyopathy. So this is a disease of the heart. So we'll go in more details now why beta thalassemia and why correcting the defect in the heart. So beta thalassemia is an inherited genetic blood disorder. Most of you have heard of people that suffering from this, they have profound anemia. So in order for those children to survive, they have to get multiple blood transfusions. And when multiple blood transfusions fail, then they need a bone marrow transplantation. Otherwise, they're not going to survive because this defective gene is very lethal for them. It affects about 1 to 100,000 people globally, and this is only a one gene disorder, which makes it perfect for us because we can correct this one gene disorder. What is more complicated is if you have 1 or 2 genes that are affecting the functionality of the organ. But at least for this profound anemia, the beta thalassemia anemia, we know that it's only one gene that's causing it. The same goes for the hypertrophic cardiomyopathy. And so this is where the heart gets enlarged because it doesn't have a functional gene. So you might have heard that very young athletes, they may just drop dead because during an extraneous exercise, for example, but they never know that they had this problem. So when on autopsy is revealed that they had this the hypertrophic cardiomyopathy, so enlargement of the heart. So this is a very deleterious mutation in the heart function. So the only way that you can survive if you already know the families that carry this gene is by doing an early heart transplantation. So again, you can see now how these two are related because when you already have been born and you already know you carry this gene and this defective mutation, then you know you're going to have either bone marrow failure, you're going to have the red blood cell failure, which leads to anemia, or you're going to have this problem with the enlargement of the heart that doesn't function properly and leads to organ failure. So both of these disorders that are genetically inherited are one copy gene, and we can correct them. So again, both of them will require, in the end, organ transplantation for survival. But the ethical concerns now that really surround the genetic modification of the embryos is that mistake that I was mentioning before. So while we are trying to make the correct genetic modification, what if we do another mistake? So what if we touch another gene that's important and is normal, but now has become defective? So we cannot accept that moving forward. Unregulated genetic engineering may lead to this new form of eugenics where people might exploit this technology. So people who have money, they will go and ask that they want to create this perfect baby. I want this baby to be the super baby. But we cannot accept that because we cannot really delineate who's going to be a super baby and who's not going to be a super baby. We have to let Mother Nature take that course. We cannot create this huge genetic drifting. It's not acceptable. And the National Academy of Sciences here and the FDA has posed these really important regulations right now. So even though the Oregon University has really published this powerful paper and how much they can do and genetically correct this disorder, the FDA still is concerned that very, very strict regulations have to be applied. So you really have to identify the people who actually will need this genetic modification. So if we already know the people, so the most severely impacted people that have this genetic disorder, they can actually benefit from this because all their offsprings, for example, will carry the defective gene and will lead to organ failure. So we can save them if xenotransplantation, for example, has not moved to clinical trials yet. So this is the other option. So here is this is a picture that I always find it is very powerful because this will be the super baby that you want. You want very high Q, no baldness, perfect pitch. He has low risk of Alzheimer and other diseases and 20/20 vision. So yeah, we can have all of that. But again, we cannot exploit the power of this technology for creating the super babies. We can only apply that only to people who actually need that and can really benefit from it, because otherwise they will have to face organ failure, which is really not acceptable if we don't have enough organs right now to save their lives. So in conclusions for that, we've shown that scientists for the first time have successfully edited the genes in the human embryos, but again, those modifications were done very, very early on. So this really raises the prospect that gene editing may one day protect the babies from a variety of hereditary conditions. But as I mentioned, the procedure is still in its infancy, so we need to edit many embryos or do this many more times to really fully understand how the technology is applying so we can have those safe embryos edited. And right now, as I mentioned, this technology is very powerful for correcting only one gene mutation. We are not there yet to correct 2 or 3, so multiple gene defects. It's only applicable only if you have one genetic defect. So again, more studies need to be conducted before we can move forward with the safety and efficacy for this procedure. So I leave you with a question that, knowing what you heard here today, if it were you and you were facing organ failure and you knew that you had a genetic defect, what would you do? Would you decide to genetically modify the embryo so that your offspring is going to be healthy, or would you use xenotransplantation so that you can save your child's life? So that's the big question. What would you do? So thank you very much again. I really appreciate you for sticking around, hearing about it. And also I would like to thank the Science in the News for the opportunity to present these two really important subjects with you today. So any questions are welcome.